All right. So thank you for inviting me to speak today. We're all in this era of speaking into webcams, and it's always better to be able to see the people you're talking to, but this is the best we got these days. So regardless of anything else, the title of my talk is appropriate. It is Time for Time, and in this acronym, TIME, TIME stands for the tumor immune microenvironment. So when it comes to cancer, immunology is what it's all about today, and it's what we're going to talk about. First, a few disclosures. I have been funded by a variety of industrial collaborators, including Bristol Myers, Leica and Aperio at Leica, Histolix, which is a startup company trying to do slide-free histology, Pfizer and Agendia. None of these has actually paid me, only funding for the research projects, with the exception of I am getting an honorarium for this presentation today. So that's my full disclosure. My objectives today are, and I've tried to keep them straightforward for you all and not get too complicated, but the tumor immune microenvironment is complex, and so this will just be sort of an introduction into what we know about the tumor immune microenvironment today and what we're studying for the future. I have a focus, which has been the focus of my laboratory, on experimental pathology and developing next-generation mouse models of human cancers, and maybe a few of the new technologies that I see in very common use in research today, but very rapidly, I think, translating into clinical diagnostics. And then the focus for me scientifically is to really attack some of the biggest problems. I study breast cancer, so you're going to hear mostly about breast cancer today. And the three biggest problems, at least in my opinion, when it comes to breast cancer are treatment of metastatic disease, reducing the overtreatment of indolent or non-lethal disease, and that's the result of sort of this failed promise of early diagnosis, where we all believed that mammograms were going to dramatically reduce the impact of breast cancer the way pap smears dramatically decreased the impact of cervical cancer, but it really has not lived up to that promise. Finally, I hope that some of you will take this as a conceptual framework for how to build teams and really do team science to tackle questions that are beyond the scope of the old way of doing research, where a single lab and maybe a single gene or a single gene pathway was being unraveled in a lot of detail. There's a lot of value to that, and certainly that has been important to the way we think about cancer, but taking a bigger picture approach is now necessary, especially as we start to talk about cancer immunology. So a lot of this starts from this important paper. I mean, obviously, even before this paper, there was a lot of research in the area of tumor immunology, but this one really got the ball rolling again, where Jerome Galan was able to show that just taking small biopsies of colon cancers, you could predict the outcome of patients based on the infiltration of T cells, those are the CD3 cells, and whether those were outside or infiltrating into the tumor. And in this case, also looking at CD45RO as a second marker, and using those two in ratios could separate even if you look at panel C at the bottom, even the early stage disease, you could find the ones that were going to behave very badly versus the more common good behavior lesions, sort of in this very well separated Kaplan-Meier plot. So that started us thinking, and I just want to go back a little bit and get everybody on the same page of where we are in cancer today. So a few years back, we realized that lung cancers could be stratified based on EGFR mutations. This was a new revolution. These EGFR mutations predicted response to specific therapies, and I've listed two of them there. And this gave rise to the question about if we have better sequencing technology, and it's become cheaper and cheaper to sequence the cancer genome, or any genome for that matter, maybe we should be looking at that for sort of all patients. And if we look at cancer genomes for all patients, we can take a different approach to the way traditional diagnostics and therapy decisions were made. So traditionally, we would diagnose a cancer in a certain organ system. That organ system cancer would go through a series of clinical trials to figure out what the best cancer therapies are. It was sort of done as an empirical, or a series of empirical experiments to determine what were the best therapies. But here, the NCI established what was called the match trial, with the idea that you could look for specific mutations that would predict a response to a specific therapy, and you could do that regardless of where that tumor originated. So an EGFR mutation in the lung or an EGFR mutation in any other location would give you a target for therapy regardless of pre-existing known sort of efficacy in that organ system. So this is a very simplistic kind of approach, actually, as complicated as it is, because it's still one gene at a time, one gene relating to one drug at a time. Doug Hanahan and Bob Weinberg published in about 2000 sort of a subway map of the hallmarks to cancer. And if you think of cancer here, and EGFR is on this list, I don't have a mouse to point at it, but there's other genes on this list. Some of them are druggable, some of them are not. But if you think about cancer just in terms of the subway map from the 2000 era, we think that it makes sense to look at one gene and one therapy, regardless of whether that cancer arose in the colon, the lung, or somewhere else. But even Hanahan and Weinberg have recognized, and so in about 2011, they published an update of the hallmarks of cancer. And you can see now they've added quite a few additional and very important hallmarks here. So on this list now, some of the same intrinsic cancer cell pathways, such as sustaining proliferation, so proliferation signaling, are still there, but some others are now here on this list that I think we should make note of. So avoiding immune destruction, so immuno-evasion, tumor promoting inflammation, the microenvironment is here. And all these things really mean that our old way of thinking about cancer was too simplistic, and we need to be thinking much more about cancer in its microenvironment. So here's a very simplified cartoon of that, just introducing you to some of the cast of characters, and you all know this pretty well already, but it sort of illustrates that with just an oversimplified view of two different active T cells as well as T regulatory cells, dendritic or antigen presenting cells, macrophages, the myeloid-derived suppressor cells, and the NK cells. Again, I could go on, but that introduces this panel now of 7 different new cells that we need to be looking for and trying to interrogate their interactions with tumors. So the other big boost to tumor immunology has been the great success of a new class of drugs to specifically target the immune checkpoint PD-1, PD-L1. And this is just a cartoon of that, illustrating the immunotherapy for cancer and how these drugs work. It's oversimplified, of course, but some cancer cells, and we found this first in lung cancer cells, have figured out how to fool the immune system by expressing this PDL1 antibody, programmed cell death ligand 1, which interacts with the T cell and tells the T cell, hey, if you're recognizing me, you're recognizing self, and you need to just calm down and get out of here. And if you block that interaction, and we've got now drugs from 4 different companies, which are antibodies either against PD1 or PDL1, they all perform about the same. They block this interaction, so the cancer cell's no longer able to tell the T cell that there's nothing to see here, and the T cells then will attack the cancer again, and this can have great effect. So many of you know, a few years ago, Jimmy Carter had melanoma metastatic to his brain and was treated with an anti-PD1, PDL1 therapy, and has had a durable response. He's now cancer-free, and he remains such, and has done very well. So if you look at the PD1, PDL1 checkpoint inhibitors, there's really still, if you took all comers of patients with metastatic disease, only about 2.2% complete response. Partial responses may be closer to 20%. And so this has resulted in a big deal for us in the clinic, trying to segregate patients based on their tumor characteristics and expression of PDL1 on tumor cells or on non-tumor cells in proximity. And then as a surrogate for tumors that have a high neoantigen presentation possibility, we look for total mutational burden. So tumors that have a higher total mutational burden make more tumor antigens or neoantigens that don't look like self, and those patients are also better candidates for this PD1, PDL1 checkpoint. So in pathology, we spend a lot of time trying to figure out how to score the immunohistochemistry, and I won't go into all the details. This is just one of multiple guides. This is for head and neck squamous cell carcinoma, put out by Dako, who makes one of the 2 main antibodies used for this purpose. And you can see that it takes a 69-page document to educate a pathologist how to interpret the IHC for PDL1. So this, I think, is still a work in progress. But I wanted to ask a more specific question about how might we improve therapy for PD1, PDL1 therapy. And so here, I like to go back to experimental pathology, and it's hard to do these studies in cell culture. You really need a mouse, and you need a mouse which has an intact immune system. So many of the models that we use are human xenografts in mice, or more recently, the so-called PDX mice, which are patient-derived xenografts. All of these are in mice with very deplete, immune deficient context. And so my interest has been for the last 20 years to try and improve our mouse models that that have intact immune systems. And you know, Ralph Waldo Emerson knew about this back in the 1800s and said his often misquoted witticism about building a better mousetrap. It was actually building a better mouse, and the world will be a path to his door. And that's certainly been the case for me. My lab built a better mouse. We built this mouse in 2005, or published it in 2005, where we looked at 2 very closely related cell lines that we derived from transgenic mice, the so-called MET1 and DB7 cells. And these are now transplantable, so you can use them with all the convenience of xenografts, but they're transplantable into immune intact mice because they're syngeneic with those immune intact mice. Many of our mouse lines that we use are congenic mouse lines, and so they are like identical twins throughout the entire colony. And therefore, they will not recognize as non-self tumors, which were derived from syngeneic animals. So this model system has been used by lots of investigators, and I just recently looked it up, and there are 89 PubMed Central articles referencing this paper and using these cell lines. And these are, we will provide these free to anyone who asks for them. So these are free to you to use in your laboratory. If you want to study the details of a breast cancer line that you can study with Immunology and really drill down on specific questions about metastasis, since these are divergent with respect to metastasis. So a similar line, a newer line that we published more recently, this was published back in January of last year, is an ERB mammary cancer model. This uses the so-called NDL transgenic mouse, which has a exon deletion programmed into it. So it looks very much like one of the common human isoforms of HER2 expression, which is known as the delta 16 isoform. And this one uniquely, again, transplantable into syngeneic mice, but this tumor cell line expresses PD-L1 in the tumor cells. And so PD-L1 is uncommonly found in breast cancer, but is sometimes found in breast cancer. And when it is, you'd have the opportunity to ask, what is the response to PD-L1 therapy? And then furthermore, how can we improve that therapy? Again, this cell line is freely available to investigators that want to use it. And even though we just published it back in January, there are already 14 publications in the literature using this cell line. And I invite you to inquire with me later if you're interested in obtaining this cell line. Again, we're happy to share it. So in collaboration with Kathy Ferrara's lab here at UC Davis, and she's now moved to Stanford, we asked and she's an expert in high-intensity focused ultrasound as a tumor ablative therapy. We asked whether thermal ablation using high-intensity focused ultrasound would release antigens and increase the efficacy of antigen presentation and therefore the immune response to the tumor. And we did this with and without anti-PD-1 therapy. You can see we used this timeline in the middle where we transplanted the cells. 3 weeks later, we performed a simultaneous administration into the tumor of CpG, which is the toll agonist. It's C and G DNA recognized by toll receptor and providing activation itself, along with anti-PD-1 therapy and along with ablation. And interestingly enough, to our surprise, we found that you did get a response. And so these are the green and red curves that you see at the right. And you got that response both in the tumor that you treated as well as the tumor transplanted on the opposite side of the animal. That tumor on the opposite side is our way of measuring what's called the abscopal response, but it can also be thought of as an experimental version of a metastasis. And of course, in patients, it's not so much the local cancer that we have a problem treating, but the metastases. And so immunotherapy, to be effective, should be effective not just against the local therapy where we inject and ablate, inject the CpG oligodeoxynucleotides. It should also be effective at the contralateral site. So you can see we were a little disappointed that we saw, in some ways, better contralateral response without the ablation, and we wondered about this. But what undaunted, we went on and asked whether we could improve this with a different schedule. And so here, instead of doing everything at the same time, if you look at the timeline, you can see that we started with the transplant. We did CpG injection along with anti-PD-1 therapy for another week, and then came in with the ablation. And in this context, the combined ablation in the contralateral site gave us almost complete eradication of both sites, but now complete eradication of that second and even third location of experimental metastasis. And so what we learned from this is that you really need to prime the immune system to be ready to recognize new antigens, and then come in with antigen release for those antigens to be presented, at which point you get very good immune tumor ablation of metastatic sites. So this has been published, and I invite you to look at the details in the publications that are listed here. This is Kathy Ferrara's group just before she moved to Stanford. So what about humans? Can we do the same thing in human context? And so here my collaborators at UCSF, Michael Campbell and Laura Esserman, asked whether we could, in the context of the I-SPY trial, and for those of you who don't know about the I-SPY trial, this is an adaptive therapy trial which has as a surrogate endpoint the efficacy seen with respect to partial or complete pathologic response in a neoadjuvant setting. So rather than having to wait for 5 or in breast cancer patients even 10 years to know whether you had an effective therapy, we can get a readout immediately at the time of surgery after the neoadjuvant window. And patients, it turns out, who have complete pathologic response at the time of surgery have a very good long-term prognosis. So those two things correlate very well. And so having that very rapid ability to evaluate new therapies gave us the ability to begin to study this. And so we have been looking at this in the I-SPY trial, where adding pembrolizumab to conventional paclitaxel, along with the doxorubicin cyclophosphamide in a neoadjuvant setting, is giving us some ideas about how to add immunotherapy to our current breast cancer. And because we have biopsies, you can see at the bottom the red arrows depict biopsies we have both before, of course, for diagnosis, but also on therapy, we can see the effect on the immune system in the context of this neoadjuvant immunotherapy. So in order to study that, we've developed some new tools. We've used the Akoya Vectra multispectral imaging analysis system in order to try and do a better job of characterizing the immune response both before, during, and after immunotherapy. And so this slide shows you some of the panels we've developed. You can see we want to know where the PD-L1 and PD-1 are expressed, but we also want to know where CD8 positive T cells, all T cells, and macrophages are. We want to know where the FOXP3 cells are, and so forth. And the AE1A3 is a cytokeratin marker that marks where the epithelial cells are of the cancer, so we can assess the proximity or interaction of the immune cells with the cancer cells. And those panels, panel 1 and 2, were the first that we started with, but we've expanded this to panels 3 and 4 and 5, and I'll in a second tell you why we went to that. In breast cancer, as I alluded to in my introductory remarks, we have a big problem, which is that we've begun to diagnose more and more breast cancer with the advent of screening mammography starting in the mid-1980s. And as we had better and better penetrance of screening mammography, we saw more and more localized cancers and more ductal carcinoma in situ lesions. So these, we hoped at the time, would lead to a corresponding decrease in late-stage breast cancers. So if we catch the cancers early, we should see a dramatic decrease in finding cancers that present at late stage, patients with metastatic disease at the time of presentation. And unfortunately, we haven't really seen that. The epidemiologists tell us that many of the cancers and DCIS lesions that we are detecting and successfully treating, if we had left them alone, they would have not caused a problem. And so this is sort of the overdiagnosis part, the bad side of screening mammography. And people go to a lot of trouble to figure out how many women do we have to treat so that we actually find one woman that we have a benefit for. And I won't get into all those numbers. That would be another talk. But the answer is that you have to screen a lot of women. You have to overtreat a lot of women in order to properly treat even one woman. And so many of my colleagues say, well, especially in radiology, we found something, you called it cancer in the pathology lab. Why is that overdiagnosis? And the person then I always have to say is, who's to blame for overdiagnosis? It's the pathologist. So when we call something cancer, you have to treat it as cancer. And the question is, are some of the things that we call cancer not really potentially lethal? And if we could do a better job of stratifying those, and here I refer you back to slide 2 with Jerome Galan, if we can maybe identify those that the immune system is already kind of taking care of, we'd be able to reduce the impact of our overdiagnosis on patients and the healthcare system. So in the setting of a consortium established by the National Cancer Institute to really interrogate this question, this is the so-called MCL consortium, which stands for Molecular and Cellular Characterization of Screen-Detected Lesions. The pathologists in the group right away set as a priority evaluation of the immune system in these screen-detected lesions, whether they were screen-detected DCIS lesions or screen-detected lesions in other organ systems. We all had the same question, which is, could we predict outcome based on specific patterns of immune cell infiltration? So these, again, are the panels that we decided to develop. The process is not trivial and maybe not quite ready for clinical practice yet. It involves multiple steps. It involves scanning the image, and here just a tissue array core is shown, and you can see that the scanned image can then go through what's called spectral unmixing. This allows us to reduce autofluorescence and increase the signal to noise for each of the channels, in this case 6 different channels plus a DAPI channel for nuclei. We then go through 2 different machine learning based analyses, one to do tissue segmentation to show the cancer cells versus the surrounding stroma, the next one to segment individual cells, and then each individual cell can be phenotyped with respect to its expression for each of the markers. Those phenotypes can come back to scores. The scores can go back to scoring in the 2 different segmented regions, the tumor region versus the stroma region, and this becomes a very powerful but very big set of data. This is another example going through the same kind of segmentation with a different type of tumor and showing a very different kind of outcome. And in this very simplistic comparison of these 2 areas, I can show you one little feature that sort of makes sense to our thought process here. And so if we compare the T regulatory cell density, that's the FOXP3 marker, in the top core versus the bottom core, you can see that the FOXP3 is much higher in the bottom core, and correspondingly the CD8 T cells, the killer T cells, are markedly reduced in the bottom compared to the top. So fewer FOXP3 cells gives you more CD8 cells and more reaction to the tumor compared to the bottom tumor having more FOXP3 cells regulating that immune response, down-regulating the CD8 immune response, and having a tumor which is much less recognized by the immune system. Oops. Sorry, skipped ahead there. So T regulatory cells, of course, are more complicated. They come in different flavors. We now know that there are T regulatory cells that are educated in the thymus. There are also T regulatory cells educated in the periphery, and in tumor cells, in the setting of cancer and tumor cells, we think these peripheral educated T regulatory cells are particularly important, and that's an area of active research in many laboratories, including my own. Coming back to the I-SPY trial, we asked this question about what kind of immune landscapes do we see in breast cancer, and using the multispectral imaging and just showing you the 5 markers that were used, the 6-marker panel that we're using on 5 cases. You can see 5 different kinds of patterns. In pattern 5, those are an example of a tumor that itself is expressing PD-L1. You can see that the tumor cells are very marked with the red color, PD-L1. This is something that we know happens in a lot of lung cancers, but fewer breast cancers. But definitely we think when it happens in breast cancer, it's important. But you can see all of the different kinds of patterns you might expect. So pattern 1, almost no inflammation whatsoever. Pattern 2, inflammation, but essentially no inflammation kind of extending into the tumor cell areas themselves. So it's sort of an immune excluded pattern. And then pattern 4 at the bottom, you can see where the tumor cells are, there's also a lot of infiltration of various CD3 positive and CD8 positive T cells, as well as macrophages. So an inflamed phenotype. So all of those, we're now asking actively which of those respond to neoadjuvant pembrolizumab. And if we look also at proximity of tumor cells to T cells, we can begin to unravel which of the cells are important, which of the cells are predictive, and how do these impact this PD-1, PD-L1 checkpoint with respect to outcomes. Just a summary of those responses, where you see a higher response, you see better p-values are the larger circles, higher in the non-complete response is blue, so that's bad. Higher in pathologic complete response is red. So wherever you see the large red circle, that's predictive of a response to the pembrolizumab. This differs based on whether you are HER2 negative, HER2 negative but hormone receptor positive, or negative for all 3 markers, triple negative at the bottom, but there are similarities across all breast cancer types. These are just the controls. So PD-1, PD-L1 is one of the checkpoints, but as many of you are aware, there's a whole host of immune synapse checkpoints that might be at play. MHC1 expression has been in the literature recently, but everywhere on this chart that you see red letters, those are drugs that are in the pipeline and in development for possible immunotherapy for cancer. And this led us to think back to that MATCH trial. So in the MATCH trial, we said, if every cancer has to have a mutation, some of those mutations may be druggable. We asked, can we do an immunologic version of that? So if every cancer has to evade the immune system, are there checkpoints or immune synapses that we can target for every cancer? And so we made a short list of, from that previous slide, of markers that we ought to look for. We've been looking at these markers. Probably each of these markers deserves its own symposium with multiple talks, but I won't go into that. I'll just point out that we can mark for each of these markers. Here's a dual mark with T cells along with the OX40 marker. Here's the LAG3 marker. Here's jitter, again, marks important cell subsets. Here's VISTA or B7H5, along with CD3. Here's VISTA by itself without the CD3 cells, so you can see that there's actually a range of expression from low expressors to high expressors of VISTA. Here's B7H4, much less common, but definitely scattered cells, which are B7H4 positive here. Here's B7H3, expressed, we think, more on antigen presenting and dendritic cells, interestingly. Here's TIM3. TIM3, you kind of have to dial up the signal to see the brightest cells, and we think it's probably the brightest cells that are the most important. So all of these immune synapse pathways are part of the future, I think, of immunotherapy and biomarkers for immunotherapy. All right, so lastly, I want to tackle sort of this precancer problem in breast cancer. The precancer problem I broke down into 4 categories. The overdiagnosis problem I showed you is largely due to overdiagnosis of ductal carcinoma in situ. And so we really need to do a better job of telling which ductal carcinoma in situ is and is not going to progress. I also think that some of the highest risk invasive cancers really don't even have that detectable intraductal component. DCIS is a heterogeneous array of diseases, but they all have this increased risk of invasive cancer. And my colleague, Laura Esserman, will tell you that the evidence does not support our current standard of care. Our current standard of care is surgical therapy, often followed by radiation therapy, to completely eradicate the part of the ductal tree that harbors this intraductal neoplasm. And I really like this picture. This is a picture of DCIS kind of at its worst. It's in action. You can see that it's not a simple question to know how the microinvasion here at the bottom left at sort of between 7 and 9 o'clock is occurring. You can see that there's necrosis in the center of this DCIS, and you can see inflammatory cells scattered around the periphery. What are those inflammatory cells doing? Are they now permitting, or are they suppressing the invasion of DCIS? And so more and more we're looking at the immune system as a way to predict risk and response. And if we begin to look at the inflammatory response around DCIS, you can see that we definitively have different patterns in different DCIS cases. So the cases are listed at the top. Conventional markers like HER2 positivity and ER positivity are given at the top bars there, with HER2 being that bright blue, HER2 negative being light blue. You can see HER2 expression is very common. We're going to come back to that. Very common in DCIS, much more common in DCIS than it is in invasive cancer. ER positivity, very common also in DCIS. But if we look at these now HER2 positive over at the left edge, and we look at the inflammatory response, these have some of the highest levels of CD8 infiltration. But even these are broken down into subgroups of 3 or 4 cases each, which have stromal PD-L1, CD68 cells that express PD-L1 at the far left, versus macrophages for CD68 that don't express PD-L1 in that box 2 steps to the right. And then in between them, high CD8 cells, but very few macrophages. There are also this group at the far right that have very little inflammation, almost no T cells and no macrophages infiltrating in or around in the stroma the DCIS cases at the far right there. In the middle, here's another group that's somewhat interesting with very little immune response, but the DCIS cells themselves express PD-L1. And so at the right, no inflammation, but it's not, we don't think, because of PD-L1. But in the middle, here's a subgroup that seems to have PD-L1 expression, mitigating the immune response to the DCIS. Right next to that, here's CD68 cells. This time the macrophages are the ones expressing the PD-L1. So all of these different categories are important. We think proximity of Tregs to other T cells is also important, and we're working on that. So very quickly, these proximity analyses can be added. You know, what I just showed you was just the very top of this picture, the very top of this heat map, but the heat map can be grown out to do all this proximity analysis and interrogation of which different categories we have for DCIS lesions. Lastly, I want to introduce you to a couple of concepts with regard to precancer. The Precancer Atlas project is funded by the Moonshot Initiative at the NCI. Here we're looking across different tumor types and asking whether we can characterize those precancers with respect to risk. Some of the sub-aims of the Precancer project involve the precancer tumor immune microenvironment 4.2 that I was just talking about, as well as just trying to get an archive of the histology and phenotypes that we interrogate in this cohort. And so just some of the very preliminary data, you might ask about ductal carcinoma in situ and the clonal evolution of lesions across space, and we've been studying that. In addition to gene expression analysis. So just like we can interrogate the immune cells in the histology, we can also interrogate the immune cells as evidenced by the gene expression from the bulk RNA expression from an area of tumor. So lastly, how do we study this back in experimental model systems? So my lab has worked with the so-called MINO model that we developed, which stands for mammary intraepithelial neoplasia outgrowth. Again, these are transplant model systems of complex tissues in an immune intact animal. And we found that you could have progression consistently from precancer to invasive cancer in this model system. Some of these are metastatic and some are not. They segregate, and it gives you a unique opportunity to study those precancerous lesions that are the most important versus those that are the most innocuous. In another model system, the STAT1 knockout mouse, which was made as a way to reduce natural killer T cell function. There's a nice Cell article from the Schreiber lab that points out that these STAT1 knockout mice are susceptible to obligate intracellular pathogens like Listeria challenge. But it turned out these mice also developed mammary tumors, and they developed a unique pattern of mammary tumor that we think is very analogous to age-related ER-positive breast cancer, which is the most common type of breast cancer, and also despite an overall good prognosis in the category, still the #1 cause of breast cancer death. And we asked whether this was because of the natural killer T cell defect associated with a difference in inflammation, whereby you might hypothesize that women as they age have reduced ability to be effective at immunosurveillance or immunosuppression of these age-related ER-positive breast cancers. And I invite you to look at this paper in detail. Very interestingly, it showed that the tumor cells express chemoattractants for macrophages, but also express chemo repellents for macrophages. And this resulted in macrophages that come close to the cancer, but then are not able to attack the cancer or prevent its spread because of the chemorepellent effects. And those segregate into extracellular vesicle fractions and soluble fractions, which are these dot plots that are very small down at the bottom in panels G, H, and I. but I invite you to look at the paper for more details. So a few clues about how DCIS might progress come from HER2. I think HER2 expression, it turns out in DCIS is seen in at least 35% in this case, compared to invasive carcinoma at only 12%. So that, I think, provides a clue that HER2 might be recognized by the immune system and might be one of the ways that the immune system prevents DCIS, at least HER2 expressing DCIS, from invading. And so we asked a simple question about how might a DCIS evade the immune system? And we think the answer is at least partially related to misexpression of isoforms of HER2. And there are different isoforms. This is sort of less well known, but is known in the literature that this pattern up at the very top, where you show that there's only detection by the green antibody, the intracellular domain of HER2, but there's complete loss of the extracellular domain of HER2. And we hypothesized that loss of the extracellular domain of HER2, while it still gives you HER2 signaling and oncogenic potential, reduces the ability of the immune system to recognize that HER2. And additional patterns were seen. You do see some full length, apparent full length, but you also see partial losses that can be subclonal, and you can see gradients of loss of the extracellular domain of HER2 in that pattern 4 at the bottom. And that we hypothesize may be due to post-translational cleavage events in HER2 that are very common. So going back to the mouse, my colleague Josh Schneider at Duke was able to model this in what's called the Cranebow mouse. And this is unpublished data, but we hope it'll be published soon and you'll be able to reference the full paper. But we were able to engineer this mouse to express the different isoforms in the same mouse and then ask about the outcomes of those mice. And very interestingly, the P95 loss of extracellular domain does show a very different kind of immune pattern, immune reaction, and shows much more invasiveness. If you do single cell RNA sequence analysis of those epithelial cells and fibroblasts, you can see that there's a much greater impact of the immune system and the fibroblast in those extracellular domain loss. And even at the very earliest stages, these P95 lesions are the most invasive and show the least containment by the stroma and the immune system. So overall, this gives us some clues, and I'm just going to wrap up here because I'm getting near the end of my time, that we may be able to reclassify DCIS in part based on the immune recognition of DCIS. We may furthermore be able to treat DCIS with an immunotherapy that would encourage the immune system to continue or increase the immunosurveillance to prevent that emergence of an invasive lesion at the stage of DCIS. And that's sort of the goal. I hope that we get there. I think you're going to see these tools that we're using in research implemented in the clinic in very short order, in part maybe to just begin to interrogate response or prediction of response to specific immunotherapies and drugging the immune synapse, but also maybe to reclassify and re-diagnose, change our classification schemes for invasive cancers as well as in situ lesions into those that are immune evasive versus immune non-invasive and kept in check already by the immune system. And this, I think, will be an important part of the future of pathology diagnostic. I hope that I've convinced you that teamwork is important here. I talked about many of my collaborators, all of them with unique skills and subsets of expertise. We may all be blind men, but we're all we're blind men and women, but we're also very good at working together and communicating. And so at the end of the day, hopefully we can get a better picture of the elephant. The pathologist has to be involved in many of the steps along the way, and I hope I've convinced you of that it's one of the roles of pathologists to put the big picture together with the gene expression, the cytokines and immunology, the flow cytometry, and put that in the context of what's going on, where the rubber meets the road, at the point where the immune cells are interacting at the invasive front of the tumor, and how effective or non-effective are those. So in summary, the major problems in cancer, including better treatment of aggressive and metastatic disease, reducing overdiagnosis and overtreatment disease, and finding real precancers very early, these nascent lethal precancers that are defined, at least in part, by a very early evidence of immune escape, as well as immune stromal interaction, are possible using some of these new technologies for research and translation. I showed you a lot of multiplex immunohistochemistry, sort of a blend of flow cytometry and conventional histology. I showed you some single cell RNA sequence data, and that's becoming more and more important in research and giving us more targets to handle these. But as you saw, this becomes a very big picture, big data problem, which requires new computing and informatics tools to handle both of those. And it takes a team of the blind scientists and a chance to put them together to make meaningful progress. So with that, I'll acknowledge all of my collaborators and co-investigators, as well as the funders and the industry partners that have made all of this possible. And I'm happy to take any questions.